Hi there, my name is Mateo Etcheveste, an undergraduate working with the UCSC Genome Browser, and today I will be using BLAT to compare the Marburg virus genome to UCSC's display of the Ebolavirus genome assembly. The Marburg virus is related to the Ebolavirus, both in the family Filoviridae. Thus, in this example, I will illustrate how we can use BLAT to compare conservation between the two viruses and discuss why certain parts of the two viruses are conserved and ways to observe this. Let's start by first opening the UCSC Genome Browser main page by typing in genome.ucsc.edu in our search bar and clicking on the link titled Genome Browser. From here, we can see a list of all the different organism assemblies currently available. If we scroll all the way down to the bottom under viruses, we can locate the Ebolavirus assembly. Clicking on this will refresh the page to the Ebolavirus assembly. From this screen, clicking on Go will bring us to the Ebolavirus's main session window. This will be our home screen from now on. Here on our main session window, we can first observe a few things about the Ebolavirus assembly. The genes expressed by the Ebolavirus and the purpose each gene serves. By clicking on the gene link, 160 virus sample strains and the conservation amongst them, and various other datasets that can be packed or unpacked. While we can immediately dive into the conservation analysis, let's first begin with the BLAT search or comparison between the Marburg and Ebola viruses. If we look at the top blue bar under Tools, we will find the BLAT tool as our first choice. Clicking here, we will be brought to the BLAT tool main page, where we can tweak the options and enter our template DNA, RNA, or protein sample into the type box. For this analysis, I will be using a Marburg virus genome from GenBank, which can be found in the description below. After copying and pasting this genome template into the box, we can tweak our search preferences or just simply click on Submit. After clicking on Submit, we are brought to the BLAT search result page. At first glance, we can notice most matches of roughly 20 or fewer bases, but the best match is at the top of the list, roughly 1,300 plus bases. If we click on the associated browser link, we are brought to the exact site of this match within our main session. Now that we are back to our main session, let's click on the top browser link, zoom out by 3x, and make a few observations. In the following session window, we can see beneath the blue Ebola virus the NCBI gene strand, our template BLAT match strand of the Marburg virus we searched for. In this session, we can see that the matches are two colors, purple and red. Clicking on the short BLAT matches, we are brought to the color key page. Here we can learn that purple is the sequence that extends beyond the end of the alignment, and red the sequence and reference genome that have different bases at this position. Clicking on the back arrow in our browser window, we are brought back to the main session. Knowing what each color denotes, we can see that a huge portion of our Marburg virus matches the Ebola virus genome, but varies in bases. If we zoom in on a particular section of red, we can directly observe this variation. The bases shown against the gray background of our BLAT Marburg virus probe, aka your-seq, represent bases that do not match the Ebola virus genome. Changes in the first and second nucleotides of a codon result in entirely new amino acids. On a similar note, if we scroll down to the bottom of our 160 strains comparative genomics data track, we can directly see amino acid variations in play among Ebola strains in these same amino acids. This can be demonstrated by the highlighted amino acid track. Using BLAT, we can see some of the similarities between the Marburg virus and Ebola virus within the L gene, an important part of the virus's functionality as it encodes for the virus's transcriptase, integral to the virus's ability to replicate and survive. This also supports the idea that these areas would be the region most likely to match, since Ebola virus and Marburg virus are in the same family and replicate and spread in the same ways. Now let's move on to our latter observation, conservation within the Ebola virus genome. As we just saw, variation or conservation of amino acids amongst the 160 different strains is higher in certain areas than others. Again, this can be seen by scrolling down to the bottom of our 160 strains comparative genomics data pack. However, another dataset we can use to observe conservation is the Philo-P dataset, which can be found near the top as a small varying thin pink line. The pink color in the browser indicates that the data extends beyond the maximum value displayed in the track. Before progressing any further though, let's click on the zoom out 100x to be brought to nearly the full Ebola virus genome assembly. To see this example of gene conservation, let's highlight and zoom in on the gene VP24. Doing so will bring us to this session window. Here we can observe the coding region of the VP24 gene, as well as the specific amino acids that it encode for this protein. Within the upper middle of the session window, we can see the Philo-P score. Philo-P tracks and compares the conservation of 158 different virus strains in addition to 2 Marburg virus strains. Marburg is included in this dataset because it is related to Ebola. However, in order to directly observe gene conservation, we will need to zoom in and be able to directly see the varying Philo-P scores. To do this, let's right-click on the left tab next to the Philo-P track and click on Configure Philo-P. Doing so will open the following window. Let's make a few adjustments to modify the track to make the data more apparent. First, let's change the display mode to full, allowing for a larger, more readable graphic display. In addition, let's change the track height from 50 to 100. Next, let's change the vertical viewing range minimum from -3.107 to -5, and the max from 0.934 to 5. Finally, let's click on Apply and observe the changes we just made. Notice how our Philo-P dataset has grown dramatically and is now presented in a lot more detail and user-friendly format. Now, let's observe our genetic conservation. To understand the Philo-P dataset, we must understand the color coding scheme as well as the numbers seen when hovering over the chart. The blue color signifies positive conservation, reporting the level that each nucleotide present within each amino acid is conserved or kept the same throughout all of the strains. Brownish red, on the other hand, signifies variation or variety, signifying a possible mutation or overall variation of nucleotides amongst the 158 strains. We can zoom into a specific blue region and observe more directly genetic conservation at play. In this session window, we can see that the methionine amino acid is highly conserved, as it's the start of the VP24 gene. Before this methionine, the conservation of nucleotides varies dramatically since these nucleotides are not encoding protein nor are essential for the coding of VP24. The small orange numbers indicate insertions in one or more of the strains relative to the reference. The main point about this observation and the previous is the argument that conservation implies functionality. In areas not expressed for within genes, we find that conservation is scarce. Furthermore, even though the Marburg virus we used in the BLAT query above did not align to the VP24 region, the Philo-P conservation score indicates that VP24 is also a necessary component of Ebola. Marburg and Ebola have diverged in the VP24 region.